Hi. So, it is a dance performance, I swear. I am wearing point shoes, but you have to listen to me talk a bit before. Sorry about that. So, I'm going to tell you about SciBallet and how to use ballet to explain science. So now you're asking, why am I qualified to do this? So my name is Dr. Eleonora Moratto. I am a biologist. I am a ballet dancer, and I am a sci artist. So I think I'm decently qualified. I'm known online as the biology ballerina. And this is what sci art is. There are several definitions of it. Usually it is considered just the joining of art and science. The way I like to use sci art is to tell a story about science. So sci art is not a new concept. This might be the first time you hear about it. Hands up if you've heard about it before. Well, some of you, that's not bad. So it is actually a very old concept. So when we go back to before science was a separate discipline, when everything was philosophy, obviously science and art were sort of the same thing, were intertwined together. And when we think of the Renaissance, we often think of Leonardo da Vinci, famous polymath, who was proficient in several things. Art, he's known as a very famous artist, but he was also an engineer and a biologist. So very famously designed the first sketch of a helicopter, and very, very intricate anatomic studies, both of human and animals. So when we look at other Renaissance figures, so thinking Botticelli, you usually think, oh, just an artist. Have you ever zoomed in on the Primavera and looked at all the botanically accurate species that he's described? So I recommend going to Florence, seeing this massive painting and looking at every single detail. Those are all local flora. So, science, botany. Now, what about after the scientific method is introduced? So, once science becomes a separate discipline. So, let's look at the 16th century. We've got the fathers of the scientific method here, so coming together of empiricism, so experiment, from an experiment to a scientific explanation, and rationalism, so thinking of a deductive reasoning to get to a natural explanation, and bringing those two together into what today is the scientific method, so the scientific revolution. What about sci art after that? Who knows what this is? Scream it. Yeah, so this is a daguerreotype. It's the first camera. And what is this?
Microscope.
Yes. So that's the very first microscope design. And guess what? They are 200 years apart, and the camera comes after the microscope. So the very, very first observations that come from the microscope are art. They used to draw what they were seeing. Now, is that science or is that art, or is that both? So this is Robert Hooke, the father of microbiology, the person who discovered the cell. And these are his drawings. And this continues even after the first camera. So that's a study of the Thames water. It's always been dirty, clearly. So 1850, after the daguerreotype, but before we could actually take a picture under a microscope. So here we are, still art. Now let's look at the other side of the coin. Did science influence art? And the answer is, of course it did. Science is culture, the same way art is culture. So Maxwell in 1865 describes light as a wave, and he also looks at light perception and realizes that we have 3 different receptors in our eyes, so we can perceive 3 different wavelengths, and those together combined make the full range of color that we can see. And he takes the first image, the first photo in color, and there it is. So this idea of color deconvolution starts influencing art. Let's think of the Impressionist movement, which is, again, around the 1850s, 1870s in France, breaking down the light into different colors. And with post-impressionism, with Georges Seurat, so French painter, again, we're in the beautiful Institut Français, let's honor some French artists, even more precise division of the light to bring your picture. So the closer you are, the more you see the deconvolution of light, the further you are, the more the picture becomes clear. Now, I said I'm a ballet dancer, and I've only shown mostly visual arts. So, what do ballet and science have in common? Well, storytelling. So, what is the output of science today? Well, this is my first author paper that came out of my PhD. So, at some point, if you're feeling so inclined, have a look. It's essentially a story telling the story of a biological process that you've studied and you and your colleagues have looked into. And ballet usually tells a human story. You can, for example, this is a performance of Romeo and Juliet, so telling the story through the art of movement. So my question has been, can I use ballet to explain my research, my PhD research? I mean, first let me tell you about my PhD. Apart from the accidental Renaissance painting moments that we've had a few times, these are the two labs that I did my PhD in, the Senna lab and the Bodsker lab at Imperial College. And my PhD was in plant-pathogen interactions. Now, big words, let's break it down. I think all of you know what a plant is. I think we're familiar with the concept. So, how can I describe a plant, the biological process of seed germination using dance? Well, here you have a side-by-side of that performed and an actual video of the process. So this is a bean germinating, and you can see that the root, this begins with the primary root exiting and growing outside of the seed, which can be represented with the movement of the leg. And then slowly, you will see the emerging of the two embryonic leaves, which are called cotyledons. And this is fairly straightforward and fairly accurately represented through the dance movement. And you can see the plant keeps growing and these two leaves keep expanding. Then obviously the plant later on grows more and more leaves and new organs. So now, what is a pathogen? So a pathogen is any biological organism, be it an insect, a virus, a fungus, or an oomycete, that makes a healthy plant sick. Straightforward. So the pathogen I worked on is an oomycete. This is an organism that looks a lot like a fungus, but it's not actually a fungus. I'll leave you to that. It's a bit more complicated than that. But and it's called Phytophthora. So Phytophthora is the pathogen of the Irish potato famine. Pretty familiar with that. And it causes rotting of the plant and the tuber, or for other plants it infects the fruit. So this is what it looks like in action. So this is a healthy plant that has been infected and it slowly rots away. So it's quite an important pathogen to fight, and there are currently no crop protection strategies to protect against this. So once your field is infected with Phytophthora, your field is pretty much gone. And it can take up to decades to actually get the whole soil to clear out. Now, how does the pathogen find the plant? So this pathogen has these things called zoospores, or just spores. Hi. So these zoospores are often released in groups, and they can swim in liquid. So especially when there is a lot of rain, you get swimming of these spores around. But when you look at them under the microscope, if you look at your left-hand panel here, they have this tumbling motion that I've used a similar motion when I choreographed the other dancers in representing this sort of tumbling. Now, how do these interact? Well, you have a healthy plant, and the spores swim towards the roots of the plant. And if we zoom in to the roots, so imagine all of these are root cells, that is the pathogen growing inside, between the root cells. How do we use dance to explain this? So on your left, you have an actual microscopy video of the spores swimming around the root tip. And then you can see on the side that the spores stick to the plant, and then the mycelium, which is when they germinate, grows within the plant cells. So this is side-by-side comparison. Now, the big question of my PhD was, is there a way to prevent this infection from happening? Well, yes. So the main experiment I did takes a plant, infects its roots with spores, and then the plant dies. But what if we apply an electric field? We insert an electrical probe next to the plant root, and we infect the roots with spores. Well, the plant doesn't die. Guess what? It works. So can I explain this using Ballet? Let me show you.